Good morning, dear students. Today we will learn the chapter Fundamental Units of Life. What is the fundamental unit of life? Yes, cell. Today we will learn about cell. In 1665, Robert Hooke, a scientist, he examined a thin slice of cork through a self-designed microscope. What is a cork? The top of the champagne bottle, that wood-like thing is called the cork. And when he watched under a microscope, he saw a honeycomb-like structure consisting of different compartments, and this compartments is called cell. Cell means a little room. Now all living things are composed of these cells, and cell is the basic unit of life. Now let us see how a plant cell looks like. In order to see a plant cell, we'll take an onion peel and watch under the microscope. When you watch under the microscope, this following structure can be observed. You can see certain black dots or a center at the center. These are called the nucleus. And the larger part is called the cytoplasm, which is a jelly-like structure. Next is the cell in human body. Here you can see those donut-shaped cells. They are the blood cells, RBC. You might have learned or heard about RBC, red blood corpuscles. These are cell, single cell. Now next is a bone cell. Bones are made up of cells. They are called the bone cells. Muscle cells. Every muscles are made up of muscle cells. Neuron. Neurons are found in your nervous system. The nervous system or the brain is made up of neuron cell. Ovum or the egg. It is also a single cell found in the female reproductive system. Fat cell. They are also a type of cell found in human body. And last is a sperm, which is also a cell, single cell found in male reproductive system. Types of organisms on the basis of number of cells. There are certain organisms which has only one cell. They are called unicellular organisms. Certain other organisms which are having one or more cells, they are called multicellular organisms. In unicellular organisms, all the function is performed by only a single cell. Why? Only one cell is present. So all the digestion or excretion or assumption, whatever process is taking place in that organism is taken place in that single cell only, and its lifespan is very short. For example, amoeba. Amoeba you might have learned in your old smaller classes. Amoeba is a single celled organism. Bacteria and paramecium, these all are single celled organisms, and these single cell organisms are called unicellular organisms. Coming on to multicellular organisms, They consist of large number of cells. Different cells perform different function and its lifespan is long. Multicellular organism. Human is a multicellular organism, right? Cells, one cell, two cell, many cell together forms tissue. And these tissue, many tissues together forms organ. Many organs together form organ system. And many organ system together form organism. So here when a lot of cells together perform a single function, then it becomes a part of a system. Now, because large number of cells are concentrated in it, it has a very long lifespan. Examples are human, trees, animals, etc. What is a cell made up of? Every single cell do basic functions like exchange of gases, that means respiration, digestion, absorption, etc. Therefore, three main components of the cells are cell membrane or the plasma membrane, which is found outside the cell. You can see a circle there. These are the cell membrane, and at the center, which you see, is the nucleus. And as I mentioned earlier, the liquid part of the cell is called the cytoplasm. Now let us see what is cell membrane or plasma membrane. Cell membrane is also called plasma membrane or plasma lemma. Now it is the boundary of each cell which separates the cytoplasm from the surrounding. Next, it is present in both animals and plant cells and is made up of proteins and lipids. Now this plasma membrane has a particular nature, selectively permeable. What do you mean by selectively permeable? What do you mean by permeable? Permeable means it allows the movement across the cell. That means some particular substances can move across the cell from inside to outside. But selectively permeable means only certain substance can move out or certain substances can only move in. Not all substances can move in or out. Understood? So, plasma membrane or the cell membrane is selectively permeable in nature, allowing only some minerals to come inside and move outside of the cell. Now, this plasma membrane is flexible and can be folded, broken or reunited. The movement of substances through plasma membrane. As I said earlier, certain substances can move from the cell to the outward portion. Now there are two types of movement. One is called the diffusion and the other is osmosis. What do you mean by diffusion? It is a spontaneous movement of substance from high concentration region to low concentration region. What do you mean by high concentration to low concentration? Just imagine you take a beaker full of water, and you slightly tilt it. What will happen? The water moves from a region where water is there to a region where water is not there. Yes or no? So that is simply diffusion. Another example I will tell you. You just spray any perfume inside your room. You will be standing at one corner of the room and then spray it. But you can even smell it from the other corner of the room. Yes or no? So what happens when you spray this perfume? The molecules move from a region where there is high concentration to a region where there is no perfume at all. Right? So this is called diffusion. Spontaneous movement of substance from a region of high concentration to a region of lower concentration. Oxygen and carbon dioxide can move across the cell membrane. So here in plasma membrane, who all can move? Oxygen and carbon dioxide can move. I said that plasma membrane is selectively permeable. It only allows certain substances to move. So oxygen and carbon dioxide can move across the cell membrane. Now next type of movement of substance is osmosis. What is osmosis? It is the movement of water molecules through plasma membrane. So here diffusion it is a movement of substance, and here in osmosis it is a movement of water molecules from a region of higher concentration to a region of lower concentration. Where there is high, from there the water molecules will move to a region of lower concentration. We will see in detail. Let us now see what happens to a cell when it is put into a solution of sugar or salt. First of all, we'll take 3 beakers. Okay. Fill the 3 of them with water, half water, fill it half water, and then we'll name it A, B and C. In the first beaker, you will put larger quantity of salt. In the next one, you will put a medium level of salt. And in the third, you will put only a very few amount of salt. So here A is called the hypertonic solution, where salt is high. B is called isotonic solution, where water and salt are in equal proportion. And the beaker C is called hypotonic solution, where the salt is very low. Now what we are going to do, we will place cell and C in each of these 3 solutions. First one, hypertonic solution. Concentration of solution outside the cell is less than inside the cell. Understood? That means hypertonic solution, salt is greater than the water molecule. So what happens when you put a cell inside the solution? Water is high in the cell and water is low in the solution. So from the cell, water moves out, right? The cell is having a higher concentration of water than the solution. So from the cell, from inside of the cell, water moves outside the cell. And what happens to the cell? If everything is out, then the cell will shrink. Next condition, isotonic solution. If I put the cell into the isotonic solution, what happens? equal concentration is outside and inside of the cell. So because of that, the cell gain equal amount of water which is lost. Now the third condition, hypotonic solution. When I place a cell inside the hypotonic solution, the cell is having less amount of water as compared to that of hypotonic solution. So what happens? From the solution, water enters into the cell, and thus what happens when a large amount of water get inside the cell? What happens? The cell will enlarge, swell up and burst. So that is the type of solution and its concentration. Now let us learn the next part, cell wall. What is a cell wall? It is the outermost covering of a plant cell. It cannot be found in animal cell. It is only a covering found in plant cell. It is rigid. Rigid means you cannot break it. Strong. Porous. Porous means it has holes in it. And non-living structure. It is made up of celluloses, and the cell wall of two cells are joined by a layer called lamellae. Here you can see in this figure the yellow colored thing, these are called the lamellae. So cell wall basically is found in plant cell and absent in animal cell. So this is a main characteristic feature which differentiate between a plant cell and an animal cell. What is the function of a cell wall? Cell wall, as you see, it is towards the outermost part of a cell. So it provides definite shape to the cell and it provides strength to the cell. It is permeable. Permeable means it allows certain particles, not certain particles, it allows substances to pass through it, which the substances which are of different sizes, they are all allowed to pass through it, and it is made up of cellulose. The next part is nucleus. I have already mentioned in the cell, the center part of the cell, which is darkened, that you see in this blue figure, these are called the nucleus. Now nucleus is called the headquarter of a cell. Headquarter, what do you mean by headquarter? The place where everything is controlled. So nucleus is a place which directs and controls all the cellular activities. What should occur in this cell? How should it occur? All those things are controlled by the nucleus. Now we have two types of organisms, eukaryotic organisms and prokaryotic organisms, based on the presence of nucleus. In certain organisms, nucleus is not present. It is absent. And they are called prokaryote. And those who are having this nucleus is called eukaryotes. Eu means true. Karyote means nucleus. Eukaryote means they are having a nucleus, and prokaryote means they does not have a nucleus. Now this nucleus is a double layered covering. It has a double layered covering, and this covering is called nuclear membrane. Now this nuclear membrane are having certain holes or pores which regulate or controls the movement of materials in or out of the nucleus. Even this nucleus is having, nuclear membranes are having certain holes through which these particular substances can move in and out of the nucleus. Inside the nuclear membrane, it contains nucleolus, chromosomes and chromatin material. The spelling here of chromosomes is wrong. CHROMOSOMES. Yeah. So here nuclear membrane. Inside of the nuclear membrane, there is certain structure called nucleolus and also chromosomes. You might have heard about chromosomes. Chromosomes, what are they? Chromosomes are those substances which contain the information of inheritance. That means from a parent to the next generation. Okay, and the chromatin material means it consists of DNA and protein. So all these things are present inside the nucleus. Now, types of cells I have mentioned earlier. Two types of cells are there: prokaryotic cells and eukaryotic cells. Prokaryotic cells means they doesn't have a nucleus or nuclear membrane is absent. An example is bacteria. And in eukaryotic cell, they are large in size. Nucleus is surrounded by a nuclear membrane and there are more than one chromosome. Example, amoeba, human, animals, etc. are eukaryotic. Now the next part of a cell is cytoplasm. Cytoplasm is a liquid part of the cell in which other cell organelles float. What is cell organelles? Just like human beings, we are made up of different types of organ which perform different function, right? The same way cell are also having different organ. And they are not called organ, they are called organelle. Understood? So cell organelles are internal organs of the cell and they are responsible for carrying out of specific jobs so that the cell remains alive. Now a question for you. Why do viruses do not show characteristics of life until they enter in living body? Have you ever thought that viruses can only be alive when they get inside a living body? Just think about it and answer it. Why is it so? Let us see what are the different cell organelles. Different types of cell organelles are Endoplasmic Reticulum, Golgi Apparatus, Lysosome, Mitochondria, Plastids, Vacuoles and Ribosomes. Let us see each one of them in detail. Here, this is a plant cell. You can see here the outermost layer is a cell wall. And towards the cell wall you can see the plasma membrane. At the center you can see the nucleus. Towards its side you can see lysosome, which are called the suicidal bags. We will see. And towards its side you can see Golgi apparatus or Golgi complex. Since this is a plant cell, you will see chloroplast, which helps in photosynthesis. And surrounding the nucleus you can see endoplasmic reticulum. Towards the side you can see mitochondria, which is called a powerhouse of the cell, and the liquid part in which all these cell organelles float is called the cytoplasm. And the major portion of the cell, of the plant cell, is filled with a vacuole which is a characteristic feature of plant cell. That means vacuoles are absent or very small in animal cells. Now let us learn this in detail. Now here, this is an animal cell. You can see here the outermost layer is plasma membrane. That means in animal cell you can't see a cell wall. Here also you can see Golgi apparatus or Golgi complexes, ribosome. The center part known as the nucleus is also present in animal cell. Lysosomes are also present. The powerhouse mitochondria is also present. Endoplasmic reticulum is also present. Cytoplasm is also present in animal cell. So here the main two difference which you don't see is cell wall is absent in animal cell and vacuole is also absent. The first cell organelle that you are going to learn is endoplasmic reticulum. What is endoplasmic reticulum? By the name it suggests it is a network of membranes present in cytoplasm, and these are present in eukaryotic cells. These are present towards the nucleus so that it can communicate messages from nucleus to the cytoplasm. Now, endoplasmic reticulum are of two types. One is a smooth endoplasmic reticulum and then the other is rough endoplasmic reticulum. Smooth endoplasmic reticulum, as its name suggests, is made up of tubules and helps in the manufacture of fat molecules, steroids, lipids. And they does not have any ribosomes present in them, and that is why they are called smooth endoplasmic reticulum. In rough endoplasmic reticulum, ribosomes are present on the surface, which makes the appearance of the endoplasmic reticulum rough, and so it is made up of vesicles and helps in the manufacture of proteins. Functions of endoplasmic reticulum: transport material from cytoplasm and nucleus. It also provides a space for biochemical activities and also helps in the synthesis of fats, steroids, cholesterol, etc. And also it helps in the detoxification of poisonous products. So whatever poisonous products is produced, it will help to detoxify it. Now, some of the proteins and lipids helps in building cell membrane, and this process is known as membrane biogenesis. Now, second cell organelle is Golgi apparatus. It is a system of tubules and membranes bound vesicles, and they are arranged parallel to each other, stacked one on the other, and they are called cisterns. Now, what is the function of Golgi apparatus? Lipids and proteins, they are synthesized in endoplasmic reticulum and they are packed in Golgi complex. It's like certain factory where packaging is done. They also transport materials inside and outside the cell and helps in the formation of middle lamellae. Towards the left, now you can see the endoplasmic reticulum, and towards the right, they are Golgi complex. Now in the left figure, the red color thing is a nucleus, and near to the nucleus you can find the endoplasmic reticulum. and those round like structures that are found on the endoplasmic reticulum are called the ribosomes. In certain parts of this endoplasmic reticulum, these ribosomes are absent. So they are the smooth endoplasmic reticulum, and where ribosomes are present, they are called the rough endoplasmic reticulum. Now towards the right, they are the Golgi complex. You can see certain vesicles are arranged in stacks, and each stacks are called cisterns. Now, what happens? The proteins which are found in the endoplasmic reticulum, they move towards the Golgi complex, and it is packaged in the Golgi complex. Now, next cell organelle is lysosome. It is called suicide bags. Lysosomes are tiny like granules containing digestive enzymes. Since there are digestive enzymes, whatever goes inside of the lysosome will get digested, and that is why the name suicide bag. Now, what is the main functions? It helps in digestion of whatever comes inside. That means it helps in waste disposal. Whatever waste is there in the cell, these lysosomes take them inside and then they digest it. So that is why they are called suicidal bags. Now here I have written a question for you. Why lysosomes are called suicide bags? If you can answer it, answer. Next cell organelle is mitochondria. It is a rod-shaped structure present inside the cytoplasm. It is only present in eukaryotic cells. That means those having only true nucleus. It is double membrane structure. In the figure you can see where the outer membrane has specific proteins and the inner membrane is folded. Can you see? The inner membrane is folded inside, and these chambers are called cristae. Mitochondria is also called the powerhouse of the cell or storage battery. Why is it called so? Because it can store the energy. The main function of mitochondria is to produce and store the energy in the form of ATP. What is ATP? Adenosine triphosphate. So, mitochondria stores ATP, and they have their own DNA and ribosomes. Coming on to the next cell organelle, plastids. Plastids are found only in plant cell. They are absent in animal cell, and they have their own DNA. There are three types of plastids: leucoplast, chromoplast and chloroplast. Leucoplasts, they are white in color and are found in underground parts of the plant. For example, potato, they are the underground part of a plant. Chromoplast, they are red or brown in color, and chloroplast, they are green in color and found in aerial parts. Aerial parts means those found towards the above the soil. That means all the shoot, above the shoot, green color, right? What you see above the green, above the soil in green color, the stem, the leaf. Yeah, they all contain chloroplast. The function of plastids is, for chloroplast and chromoplast, they are important in photosynthesis, and leucoplast, they are the storage of materials such as starch. It is the storage of material such as starch, oil, protein granules, etc. Now next cell organelle is vacuole. These are membrane bound regions in the cytoplasm, and they are filled with water, amino acids, sugar, various organic acids and some proteins. As I said earlier, vacuoles are very small in animal cell or absent, and plant cells, they are having very large vacuoles. In certain organisms, single cell organisms like amoeba, vacuoles contain the food item. What is the function of vacuole? It stores materials. And the main important role is expelling of excess water from the cell. Now last cell organelle is ribosomes. What are ribosomes? They are minute particles consisting of RNA, ribonucleic acid. Ribosomes are main sites of protein synthesis. So protein is synthesized or protein is made in ribosomes. And also the protein is transported to endoplasmic reticulum. Now with that, we come to an end of this chapter. Stay at home, stay healthy, stay safe. Thank you. Now with that, we come to the end of this chapter. Stay at home, stay healthy and stay safe. Thank you.